Good morning or good afternoon, everybody, depending on where you are. I'm Alan Kliger, a nephrologist in New Haven, Connecticut, and co-chair of ASN's COVID-19 response team. Today is World Kidney Day, and it's also the one-year anniversary of the official designation of our COVID-19 pandemic. It's therefore particularly appropriate to welcome you to today's presentation, in which a panel of experts will discuss the use of monoclonal antibodies for the treatment of mild to moderate SARS-CoV-2 in patients receiving dialysis. Also, we'll hear the latest information about the known SARS-CoV-2 variants. These are my disclosures. So here is our agenda. As you see, we're going to hear about clinical trials in the general population, operations and the SPEED program to get monoclonal antibodies to patients, the clinical use in patients on dialysis, and finally, SARS-CoV-2 variants. That'll be followed then by a roundtable of questions and answers that you have asked. Just a couple of quick housekeeping issues. The roundtable portion of the webinar will include the questions submitted by the audience members during the registration. Handouts, including the slides, will be available, and there's the website to get those slides. And it's being recorded so that if you would like to look at it again or to share it with colleagues, you can get the recording posted on the ASN COVID-19 webpage later this week. Just a quick statement that says that the use of monoclonal antibodies for the treatment of mild to moderate SARS in patients receiving dialysis is intended to be informational and educational and should not be construed as medical advice or recommendations for patients' care. So if there are any lawyers out there, that's for you. It's for all of us, I guess. Okay. Finally, in acknowledgement of the changing nature of medical information and practice, particularly during a pandemic, let's remember that things continue to evolve and change. Okay, so let's get started. To begin with, it's my pleasure to introduce Dr. Michael Anderson. Dr. Anderson is a pediatric critical care physician and a nationally renowned expert in disaster response and health policy. He's currently a senior advisor to the Assistant Secretary of Preparedness and Response at HHS. Dr. Anderson is a key leader of the COVID therapeutics work group and has been deployed with federal teams to areas of crisis. Dr. Anderson.
Thank you, Alan. It's a pleasure to be with you here today and to represent Health and Human Services and the Assistant Secretary for Preparedness and Response. I do think it's fitting on World Kidney Day, also when marking a year of this terrible, terrible crisis. Before we get into our remarks, and you've assembled just a terrific panel to just recognize and remember those over 500,000 Americans that have died during this pandemic. We have to honor them. As Alan said, this is not over. There's still much good work that we have to do, and I think it's terrific that ASN has gathered such a terrific panel together to talk about it. So really an honor to be here, and my colleague, Lieutenant Commander David Foss, and I will give you sort of a high-level overview from how we're envisioning these therapeutics. We used to be known as Operation Warp Speed. It's now in general called the Federal COVID Response, or FCR. That is a multi-agency response combining FDA, NIH, HHS, and other resources, really a whole of government and a whole of society response. So once again, honored to be here. That's me. That's my picture. I have no financial disclosures. Please note that I am a contracted consultant to the federal government. The really important swim lane is I'm giving you my opinion as a healthcare leader and as an intensivist. I don't speak on behalf of the federal government because I am a contractor. Likewise, everything we talk about today is not really off-label because it's under the emergency use authorization, or the EUA. And I think some of my other learned colleagues can also talk about the implications of that. So what we're going to do from the federal side is currently discuss the 3 available monoclonals, talk a little bit about patient eligibility, and I think a couple of my colleagues on the seminar will dive a little bit deeper. Lieutenant Commander Foss will then talk about our, I believe, really important effort to make sure that these therapies are getting to the most vulnerable amongst us. And we've got a program called SPEED that is laser focused on health equity, but we're always learning and trying to bring in your great work, whether it's chronic renal patients or dialysis centers or any such thing, I think is going to add to this effort. And then once again, I have been so impressed with collaboration really across the medical spectrum since we got into this public health emergency. This is a schematic of the progression of patients through COVID. Obviously, there is a lot of attention to the far 2 left chevrons. I think it is an amazing testament to the whole of the academic and pharma communities coming together that we now have 3 EUA approved vaccines. That's obviously to prevent patients from ever getting this disease. That's in these first 2 buckets positive for novel SARS-CoV-2. And I think despite some initial sort of speed bumps in the rollout, an understatement, now you are seeing a marked uptake, especially in the most vulnerable amongst us, and the percentage of patients vaccinated over 65 continues to go up. We're really talking about that middle chevron, these early symptomatic patients who have tested positive for novel SARS-CoV-2 or COVID-19, but are not sick enough to be admitted. They don't have a high O2 requirement. They're not showing elements of shock. Up until these 3 EUA approved monoclonals were out there on the market, we just really had hope and prayer that the primary care world, the emergency department world would have to send patients home and just really hope that they didn't get better. Now, if we treat patients early, if we follow the EUA, we have mounting evidence that these make a dramatic improvement in patient symptoms and in decreasing the need for hospitalization. I'm a little bit, I don't want to say nervous, but a little bit of introspection here that typically when I would give a grand rounds, I would bring you 5 New England Journal trials and 4 JAMA trials and bring you thousands and thousands of patients to show a therapy works. This under an emergency use authorization at a time of a public health emergency is really a growing snowball of information that is pointing more and more to these therapies are effective. And I think some of my other learned colleagues, I had a chance to see their slides, will start to show you the early data, but it is really early data. And maybe in Q&A, we can talk about how does organized medicine respond at a time of public health emergency? Because first and foremost, we want to do things that are safe. We want to make sure the data shows that this is an efficacious therapy. And we'll talk a little bit more about that. We're not really going to talk about remdesivir, convalescent plasma, or the other therapies for your patients when they're unfortunately sick enough to be admitted. Obviously, there are 3 monoclonal antibodies, 2 from the Lilly Corporation, 1 from Regeneron. We'll dive a little bit deeper into the hood, into those cocktails. And obviously, the whole focus of this federal therapeutics, once again, the federal COVID response, is to get these therapies to the most vulnerable amongst us so that first and foremost, we can save them from progressing and sometimes dying. And 2, our hospitals have just been overrun. And the fact that they are just either at or past the breaking point just tugs at my heart as an intensivist. So the more we can do to prevent patients from progressing is, of course, first and foremost good for that patient, but also really important for us to preserve hospital capacity. This is about the only data slide I will show you, which once again, I do with a little bit of trepidation at such a learned audience of the ASN. This is the early data below the hood of these 3 monoclonals. I will say that the BAM-EDI combination by Eli Lilly in the middle, and once again, I think you'll hear from some other colleagues on this webinar, the BAM-Lilly has gone through phase 3 trials and is now part of NIH guidelines. So NIH, up until just 10 days or a little less ago, had not put monoclonals on their approved therapies for patients in that early pre-hospital phase. The reason being is these are under EUA and they didn't feel like they had enough data. Now BAM-Lilly, the combination therapy under BAM-EDI under Eli Lilly is part of the NIH guidance, and we are confident that with more data coming in, these other two therapies will also make it their way to the NIH guidance. You can see that these are relatively small numbers, and once again, I think my colleagues will also show you more data under the hood. This is a schematic, probably oversimplification from a Nature reprint on monoclonals, but I thought it sort of drove home the potential patho mechanisms of these monoclonals. First, they bind to the to the spike protein and decrease the ability for the virus to be taken into the cell. Number two, they block membrane fusion so that the virus can't replicate. Or three, they can also then lead to what our naturally occurring antibodies do, and that is present the virus for destruction by the immune system. So three potential mechanisms of action of these therapies. This is the EUA criteria, and I think a lot of folks on this webinar, and certainly every panelist here knows these. The EUA from the FDA does not say every patient that turns positive for COVID gets these therapies. It's in really two buckets: adults and children. First, kids 12 to 18, and you can see the criteria there, qualify for these EUAs in the first 10 days. And more of the data coming in shows the earlier patients get these therapies, the better. That's why we are trying at the federal government more and more to get testing, turning positive, tied as quickly and efficiently to getting antibodies as quickly as possible. To the left is the adult guidance. Anyone over 65, repeat, anyone over 65 automatically qualifies for the EUA approval of these therapies, any one of the three. And then you can see if you're 55, between 55 and 65, you have to have one of these comorbidities. I've gotten a lot of questions over the past couple of weeks. I'm not part of the FDA process. I don't really know it well. In full transparency, are there ways to iterate and move these guidances or these guidelines to include more patients? And I certainly think the FDA is open to that. I just personally and professionally haven't seen anything new come down the pike. Once again, while I'm nervous, I'm not showing you reams of New England Journal data. I am really excited around the momentum that is building. And about 7 weeks ago, I really felt an inflection point when Dr. Anthony Fauci and Francis Collins called up the AAMC and said, get your deans and your academic medical center leaders together, because we've got to figure out how to get these therapies into more patients. Both Drs. Collins and Fauci said, the data is mounting, our hospitals are at the breaking point, these therapies are safe, and we've got to figure out A, how to publish more, and B, how to make this a much more efficient process. So when somebody turns positive, they can get the therapies as quickly as possible. I've also seen academic centers across the country. I've just listed a few here. It's not an inclusive list, but we've really been impressed with how academic medical centers have taken these therapies on and make them really a part of the fabric of their care. So it is my pleasure to introduce Lieutenant Commander David Foss, who's going to give you a little deeper dive into what we call the SPEED program, once again with the goal of getting these therapies into the most vulnerable amongst us.
Michael, thank you. That was terrific. So yes, let us introduce next Lieutenant Commander David Foss. Commander Foss is stationed at the FDA Center for Drug Evaluation and Research, where he serves as a regulatory review officer for the Office of Prescription Drug Promotion.
All right. Thank you, Dr. Kliger and Dr. Anderson. I appreciate that introduction. I am Lieutenant Commander David Foss, and I'll be discussing operations and speed. All right. Oops. There we go. Nothing to disclose, and let's go ahead and jump in. So I'm going to discuss first, I'm going to discuss the administration of outpatient monoclonal antibodies. So before you obtain monoclonal antibodies, you need to ask yourself, what do I need to do? So first, you need to make sure there's dedicated space to separate COVID patients from non-COVID patients. You also want to ensure dedicated sources of supplies, which include PPE and emergency medications in the rare chance that there is a severe reaction. You also want to make sure you have sufficient personnel. Unfortunately, dialysis centers are pretty well equipped to take on these, making pretty ideal recipients for monoclonal antibodies. Also, you want to make sure the drug administration process is set up. it can take up to an hour of treatment for the Regeneron product, 16 minutes for bamlanivimab if you use a 50 ml bag of saline, and then 21 minutes if you use a 50 ml bag of BAM-EDI. And then that kind of increases the more volume you use. So you need to calculate that. And then also it's a 1 hour post-treatment observation period. So you need to configure that and figure out how that fits into your practice. Also, I want to make sure there's a process in place for reimbursement, and Dr. Dittrich will mention that later on. And as well as making sure that you're meeting the reporting needs for the EUA. And you want to check with your parent organization to make sure they don't already have processes in place, because they may already have them. So you want to check with them, and may make your life a lot easier. So next question is, how do you obtain these therapies? So HHS ASPR currently manages the distribution of the monoclonal antibodies under the EUA. And given the current supply, bamlanivimab, BAM-EDI, and the Regeneron product can be requested via direct ordering for all sites. And direct orders of the Regeneron product and BAM-EDI are limited to 48 patient courses per site per week. And now also we ask that you really want to limit how many patient courses you request, just in order to fill your needs. If you only need 10 courses, probably only request 10 courses, don't request 40, because we want utilization rates to be high so that we can get it into as many patients as possible. And then I want to draw your attention to the right-hand side of the screen, which discusses the direct ordering process. So if you click on that link, it kind of walks you through how you can go about obtaining these monoclonal antibodies, and it also discusses the tracking requirements for teletracking. If you have any questions, please go ahead, contact COVID-19 Therapeutics at hhs.gov. And you can also contact the sole distributor, AmerisourceBergen, directly at c19therapies@amerisourcebergen.com. Now I'm going to talk about the special projects for equitable and efficient distribution, the SPEED program. So this was established in mid-December of 2020, and the goal of this was to facilitate access and utilization of monoclonal antibodies to priority and underserved populations. So this includes long-term care facilities, dialysis centers, federally qualified health centers, and correctional facilities. And the goal is to provide direct federal allocation and get it out to sites as quickly as possible. So in order to get the word out, we developed partnerships with stakeholders and trade associations. And you can see this, the website is there below if you would like to learn more. And if you have any SPEED related questions, please contact the SPEED team at covidtx@hhs.gov. So now I'm going to talk about SPEED and dialysis. So some partners of SPEED that we're thankful to have are American Society of Nephrology, who's been on since the beginning. We also partner with ESRD networks, the Kidney Community Emergency Response, and the Renal Healthcare Association. Some of the dialysis companies that we partnered with include DaVita, Fresenius, and US Renal Care. And we also work with the state departments of health. So to date, we've distributed monoclonal antibodies to over 1,100 facilities across 44 states. And in addition to the larger dialysis companies that I mentioned, also have distributed to independent centers and hospital-based dialysis units. And SPEED is always looking to grow. So if you're on this webinar and thinking that you might be interested, please reach out, go to the direct ordering site. Or if you have questions or hesitancies, please email covidtx@hhs.gov, and someone from the SPEED team can answer any questions you have, because we really want to get as many monoclonal antibodies out to the patients that need as quickly as possible. So please don't hesitate to reach out. And to date, over 3,000 courses of bamlanivimab and 6 courses of the Regeneron product have been distributed to dialysis centers. And then as far as ask for providers, we do ask that providers support referral pathways and share information with patients regarding monoclonal antibodies. And then we ask that providers administer monoclonal antibodies if their space and staffing allow. We also ask that Sites order monoclonal antibodies directly from the distributor through the website that I mentioned, and that numerous resources are available. So we ask that providers go to phe.gov and combatcovid.hhs.gov in order to learn more about monoclonal antibodies and share that information. Also, we ask that providers share experiences if they've used monoclonal antibodies and share those experiences so that other providers can learn from them, and especially if those experiences have been beneficial. And yeah, you can share them at COVID-19 at their COVID-19 therapeutics at hhs.gov. And then just to run through a couple of quick resources here for you. Baseball cards, a quick overview of the monoclonal antibodies. There's also administration playbooks that really kind of talk about the details of the administration of monoclonal antibodies. A reimbursement link, as well as general guidances. And then also office hours. So these are meetings that they're forums, open forums where you can call in and ask any kind of pressing issues, any kind of questions you have about monoclonal antibodies, and the ASPR team will help to answer them and work to get you an answer to those questions. Also, there's an ECHO mini-series, and this happens every other Wednesday, and they talk about different topics regarding monoclonal antibodies. So very educational, and I highly recommend them. And that's all I have, so thank you.
Thanks, Commander Foss. So we've now sort of heard some of the basics about the monoclonal antibodies and about the SPEED program to get these antibodies into the appropriate patients. Next, I'd like to introduce Dr. Mary Dittrich. Dr. Dittrich is Executive Vice President and Chief Medical Officer at U.S. Renal Care. She's a practicing nephrologist and also serves as President of Liberty Administrative Services and CMO for Glacier Management Services in Alaska. Dr. Dittrich gets around. She and her group have had hands-on experience in dialysis patients with these antibodies, and so she's going to talk to us about that experience. Mary?
Alan, thank you so much. And I just am so pleased to be able to be in front of you all today and share a little bit about our experience with the monoclonal antibodies, particularly, as has been mentioned now, 1 year into the declared pandemic. So the disclosures are as Dr. Kliger has described. So it should come as no surprise to anyone on the call that the population of patients with kidney disease, including late-stage CKD, but particularly dialysis and transplant, have been uniquely adversely affected by COVID-19. So at U.S. Renal Care, over 15% of our dialysis patients have contracted COVID-19 over the last year. And that's consistent with national data. So high infection rates. And the reasons for that are not surprising. Our patients have frequent contact with the healthcare system. They're community dwelling, but transit through the community and then, of course, come together with many other patients and staff to receive dialysis. In addition to a susceptibility to becoming infected, once infected, the patients have significant adverse outcomes or risk of severe disease, as we'll describe. And that includes a greater than 50% chance of being hospitalized if one of our patients on dialysis gets COVID, and above a 20% mortality rate in the published literature. I will say that our home patients are also affected, although less in our experience and in the reported literature. So what can we do? Well, Dr. Anderson did a nice job showing about where therapeutics fit in, but the bottom line is until really November, there wasn't anything we had to offer for patients early in disease, so high-risk patients before they got into the hospital. So we read with delight the study that was published in the early BLAZE 1 trial that was published in the New England Journal of Medicine in late October, and an EUA was obtained in November. I'm not going to recap the data here, which was nicely summarized. Suffice to say, it's evolving. There was a press release yesterday with new data. But the point I would like to make is that in the bamlanivimab trial, which has released phase 2 and now phase 3 data of over 1,000 patients, there were less than 10 patients with late-stage kidney disease and dialysis in that trial. And similarly, in the Regeneron trial, there were very few patients with kidney disease in the trial. So we are really operating on the limits of data here, especially in patients on dialysis. So I just want to reiterate the high-risk criteria. So again, these have to be outpatients, and our patients are eligible for this therapy based on their chronic kidney disease. But again, it bears mentioning our patients often have multiple comorbidities that put them at risk for adverse outcomes, which is why I think we see the outcomes that I've described of mortality and hospitalizations. Our patients very often have comorbidities, including obesity, diabetes, hypertension, and cardiovascular disease. Dr. Anderson mentioned, and I'll reiterate in my lessons learned, that we were pleased to see the updated NIH statement at the end of February, which did now recommend the use of the combination bamlanivimab-etesevimab for, again, the same group of patients that were referencing outpatients with a recent diagnosis of COVID-19. And they reiterate, and you'll hear from all of us, that early treatment, especially as you understand the mechanism of action, which is these, antibodies bind the spike protein and prevent attachment via the ACE2 receptor and entry into the cell with replication. So early treatment is important. As the literature has evolved, so has this professional guidance, and the ISDA continues to recommend against the routine use of bamlanivimab for ambulatory and, of course, hospitalized patients. But they do state in groups of patients like those with kidney disease who we know are at increased risk, consideration can be given to the bamlanivimab treatment, of course, recognizing informed decision-making between the patient and the provider should occur. So just to summarize some of the data, there's early. Dr. Anderson stated that we're early in the data evolution with these therapies. But there's a suggestion for sure that it can mitigate severe diseases evidenced by lower hospitalization and ED visits, and there may be a mortality benefit as well. We didn't go into the safety data, but both in the published literature and in our experience, these are well-tolerated therapies, although there's also a documented risk of infusion reactions, hypersensitivity, and anaphylaxis. So being prepared to manage those complications is an important part of preparing to give these medicines. I'll reiterate there's very little data in dialysis patients, but I do want to emphasize, as Lieutenant Commander Foss and Dr. Anderson iterated, dialysis facilities are uniquely well positioned to administer this therapy. So we have high-risk patients that regularly come into our center for a prolonged period of time, allowing us to administer the therapy and do the required observation. We have vascular access, which is often a barrier. We have staff trained in emergency management of complications. But early on, we had trouble obtaining the medication. We were having to go to individual state health departments. So this collaboration with the SPEED program markedly accelerated our ability to get this therapeutic to our patients, and we appreciate that partnership very much. This drug we are obtaining at no charge, so the government providing this drug, and so we do not bill for it, but questions come up about reimbursement. So again, there's no charge to the patient, the facility, or the organization, but there is an allowable administration fee to compensate for administration time and supplies of $310, and details can be found on the referenced website here. So how did we approach providing bamlanivimab? So very early in the pandemic, we recognized we need to partner with our physicians to treat COVID-positive patients. And we initiated a series of calls in which we had infectious disease and other specialists come on. And so in November, we presented data on this therapy and continued to present data on this therapy as it evolved with our physician partners. And they then reached out and expressed a willingness, many of them, and a desire to administer these therapies. And as I'll mention, physician collaboration is key to being successful in administering these. In addition, we have a dashboard where we follow where our hotspots are or where our cases are rising. And then my office would do directed outreach to physicians in areas where we saw cases going up to see if they wanted to administer the therapy and facilitate that. So we started with a limited number of facilities, and now we've rolled it out so any of our facilities can give the therapeutic once they've been trained and prepared. And we did staff education as well. There were a couple logistic things we had to get in place, and I will talk a bit about the need for rapid testing. You've heard again and again how early treatment is mandatory to get the desired effect, which is why it doesn't work in hospitalized patients. We had to augment our ability to turn around testing quickly. Policies and procedures had to be drafted. We retrained our staff in infusion reactions and made sure we had emergency meds in the facilities and people knew how to use them. We had to put processes in place to do the mandatory reporting. What were some of the challenges we faced? There was some reluctance on behalf of our physicians to use a therapy that was under EUA and not FDA approved, and they hadn't had experience in this. Nephrologists have used monoclonal antibodies, but not this particular one. A big challenge for us was logistics. So these are COVID-infected patients treated, as is true throughout our community, on ISO shifts. And those are often end of day, and that staff is burdened by taking care of a complex group of patients. And so we didn't lightly undertake asking them to take on another task. And we did a lot of education about why we were asking them to do more, but that was a barrier we had to overcome. And I'll talk a little bit about timing of administration. Early on, we followed what was done in the Blaze and Regeneron trials, where the therapeutic was given after dialysis. So our patients on an ISO shift at the end of the day had a 4-hour treatment, an hour-long infusion, followed by the requisite observation time, so over 6 hours in the facility. And that was a huge barrier to uptake. It was not acceptable to patients, and our physicians struggled with that aspect as well. And I'll talk about how we resolved that. We required our physicians to do consent, and that was a challenge because, as you know, they're not often in the facilities or often not in the facilities, and we needed this to be timely. I'll talk again about some solutions we found there. We had to put an ordering process in place. There's a requirement for certain infusion sets that we had to get to the facilities. And then the medication is diluted. Both of these medications are diluted in saline, and we had to have the right type of saline bag. So those are just some logistics. So what did we see? Was it worth it? I guess is what I'll say here. And this is some of our data. So I'm presenting early data, earlier than we would typically present data, on our first 40 patients. So we started administering the drug in December, and we have 40 patients that we followed for at least 30 days. What you see here in the blue bar is matched controls. So we identified a group of COVID-positive patients not treated with bamlanivimab, but matched on diabetic status, age, race, and gender. And you can see, as I reported, that within 7 days, over 57% of those patients were hospitalized, whereas in the bamlanivimab group, 6. So small numbers, but significantly fewer relative bamlanivimab treated patients were hospitalized. And similarly, out to 30 days, we still saw this decrease in hospitalization. So early data, small numbers, but impressive, I think. I'd like you to consider 2 things here as I go to the next slide and talk about how we interpret the data. So first of all, these patients have a very high risk of hospitalization, and the hospitalizations occur early. So 57% of patients that were hospitalized were so within the first 7 days, and then the subsequent 23 days, an additional 8% or so were hospitalized. And the second thing is, as I mentioned, small numbers overall. So we were enthusiastic about our early data, but interpreted it with caution. And that's because there's inherent bias in this data, which I'll describe to you. So there's methodologic issues with how you have to give this medicine to get it in early. So the EUA dictates that it's outpatients that are not hospitalized and have tested positive for COVID-19. And there's logistical issues, frankly, with getting this drug in quickly in our patient population. So typically the patient presents for dialysis or reports symptoms, a test is ordered, the results are delivered, which sometimes takes 48 hours. The physician is notified, the patient is rescheduled to a COVID shift, then consent obtained, and we give the medicine. So some logistic challenges there. In our company, in our organization, that took typically 4 days. The mean time between a positive test and administration of the therapy was 4 days. Well, I already told you that the vast majority of hospitalizations happened early here. So that's statistically described as immortal time bias, which is basically when the follow-up period includes a period of time where participants in the exposed group cannot experience the outcomes. That's called immortal time bias, but basically means you have to stay out of the hospital to even get this drug. And that may bias our results, and we recognize that. But how do we interpret, or what conclusions do we draw? Dr. Anderson and Lieutenant Commander Foss encouraged us to share our results, and we're doing so and appreciate this forum in which we do it. But we just advise caution in looking at this because of all the factors I've described. But we believe that our unadjusted and both our unadjusted and case control approach, evaluating what we term efficacy, suggest improvement in hospitalization rates. But there is this immortal time bias I noted. I didn't show you this data, but there's a suggestion of a mortality benefit with a substantial reduction in hazard ratio of almost 80%. But again, very small sample size, and so we can't estimate statistical significance. And again, we have not followed these patients that ended up being hospitalized to make a determination about either the mitigation of severe disease or long COVID. So we don't have data on that. So what lessons have we learned? We're about 2 months into this. And first, I will emphasize that I was an early adopter, and I really wanted to make this therapeutic available. And the collaborations across our industry with my colleagues, the CMOs at other dialysis companies, the team at FCR, used to be called Operation Warp Speed, science representatives from Lilly and Regeneron who helped us navigate that were incredible. And those collaborations were what made this possible. We know now that you have to make this easy. People are busy, patients are ill, staff are tired. And we probably overcomplicated our policies and preparations, and I think it led to some reluctance, and we've streamlined it a bit. So we developed an order set, which makes this easier. I talk about pre-recorded educational content, which was one of the best things we did. We did webinars on this, on how to administer it, how to manage complications, what the data was. And then we could distribute that out repeatedly and update it to have our clinicians listen to it at a time frame that worked for them. One important update we've made is now when on our app, we have a COVID tracking app where all our patients are screened. And when a positive result is input, our app now has a pop-up to say, has monoclonal antibodies been considered for this patient? And these are the little triggers that I think help with uptake. I mentioned that having the physician do the consent, which we felt strongly was important given the fact that this is a non-FDA approved therapeutic, was initially problematic. But when we allowed that to be done through telehealth, that made it much easier for our clinicians to obtain timely informed consent, which is key. I mentioned this timing of administration, and we have decided, along with others in the industry, in collaboration with experts and including the pharmaceutical companies, we're now giving the drug on dialysis. And I can talk a little bit about more about why we made that decision. But that has dramatically simplified administration and improved uptake of this. It's just more acceptable to patients and providers, and we believe safe and effective on dialysis. I'm happy to comment more on that if there's interest. Physician leadership is absolutely critical. We leave this decision up to our doctors. We partner with them, we put processes in place, but ultimately this is a provider decision. And as our doctors gained more experience with this, they felt comfortable with it, and our use went up. Although, as the COVID numbers came down, that's a trade-off I'll be glad to make. We've been giving less bamlanivimab recently. Mentioned the pre-recorded educational content and the fact that the data is really rapidly evolving on this. So on our calls with our physicians and on our calls with our team, we continue to update people on the literature and what we know about this. And I already mentioned that we as a community have to report our results so that we can make informed decisions about continued use of this therapeutic. And so I am a little nervous reporting really preliminary data, but have made a commitment to do so, and again, speak regularly with my colleagues at DaVita and Fresenius who are giving the therapy as well and sharing our results and experience, which I think is important. And then, of course, I'd be remiss to say that disease prevention is better than disease treatment. And we continue to believe that the best way to manage COVID-19 in our populations is to prevent it through timely and effective vaccinations. And we appreciate the advocacy from ASN and others on the call to try and get vaccinations for our patients. So I appreciate this forum, the opportunity to share our experience, which has been really exciting. And as a practicing clinician who lost patients to this disease, it feels so good to have a therapeutic option, which is looking promising to mitigate severe disease in our patients. So I'll pause now and then be available for questions at the end of the discussion.
Thank you, Dr. Dittrich. Really great talk, and there have been several questions, so hang in there, and at the end we'll ask some of them. Now, just to shift gears a bit for our last speaker, I want to introduce Dr. Pavithra Natarajan. Dr. Natarajan is with the Centers for Disease Control and Prevention's COVID-19 Emergency Response Team, and we've asked her to specifically talk to us about the variants of the SARS-CoV-2 and what we know about them and where we're headed. Dr. Natarajan?
Yes, thank you so much, Dr. Kliger, for the introduction, and good afternoon all. My name is Pavithra Natarajan, and it's my pleasure to be here today to speak about the SARS-CoV-2 variants. I don't have any disclosures to declare, and here's what I plan to cover today. So I'll speak about the emerging SARS-CoV-2 variants and the effects of these variants on transmissibility, disease severity, and vaccine efficacy. And finally, I'll cover CDC's role in tracking and responding to these emerging SARS-CoV-2 variants. So you don't need me to tell you, as you well know, the COVID-19 pandemic has affected every corner of the world. There have been over 116 million confirmed cases of COVID-19 globally. Next slide, please, and including 2.5 million deaths reported to WHO. This includes nearly 29 million cases in the US and over 526,000 deaths. So today we're going to focus on the SARS-CoV-2 variants. So viruses, as you know, are constantly changing, and this includes SARS-CoV-2, the RNA virus that causes COVID-19. These changes in the genetic code of a virus are called mutations and naturally occur over time when a person is infected. And these mutations can lead to the emergence of new variants that may have different characteristics. While a certain amount of genetic variation is to be expected as SARS-CoV-2 spreads, it's important to monitor circulating viruses for key mutations that happen in important parts of the genome. Many mutations do not affect the virus's ability to spread or cause disease because they don't alter the major proteins involved in infection. But variants with mutations that do enhance the virus's ability to spread or cause disease are able to outcompete other variants. And on that diagram, you can see that we've already mentioned the spike protein that mediates SARS-CoV-2 entry into cells. And we'll talk about this further as we talk about the variants. So multiple SARS-CoV variants are circulating globally. Several new variants emerged in the fall of 2020, most notably the three covered on this slide. So starting from the left, B117, which emerged in the UK with a large number of with a large number of mutations. This variant has since been detected in numerous countries across the world and was first reported in the US at the end of December 2020. Then B1351, which emerged in South Africa independently of B117. This variant shares some mutations with B117, and cases attributed to this variant have been detected in multiple countries outside of South Africa and was first detected in the US at the end of January 2021. And finally, P1, which emerged in Brazil. This variant has 17 unique mutations, including 3 in that receptor binding domain of the spike protein. This variant was detected in the US at the end of January 2021. So these SARS-CoV-2 variants of concern have mutations that alter the receptor binding domain of the spike protein, notably the N501Y mutation that occurs in all 3 variants, as well as the E484K mutation that occurs in the B1351 and the P1 variant. Now, it's really important to emphasize that these are the current variants of concern, but more or different variants could be targets in the future. And almost certainly you may have heard of other variants that are of interest and are being monitored closely. So now I've just got some maps to show how these variants have spread, having been reported in multiple countries. So this first one is for B117, and then next slide, we have B1351, and on the final slide, P1. This slide shows the reported emerging variant cases in the United States, and you can see that the B117 variant, so that's the one that originated in the UK, is being reported most widely, with over 3,000 cases reported to the CDC from 49 jurisdictions. And this is on the CDC web pages and updated weekly. Moving on to the effects of the variants, what are their effects and why are we monitoring them so closely? Well, first of all, increased transmissibility. So these maps show the rapid spread of B117 in England and London in late 2020, using S-gene target failure as a marker for B117. Now, not all S-gene target failures are B117, but it's a sensitive PCR marker and is being used as a proxy to detect B117 in the UK. There's lots of evidence that B117 is more efficiently transmitted compared to other SARS-CoV-2 variants circulating in the United Kingdom. For a start, UK regions with a higher proportion of B117 sequences had faster epidemic growth than did other areas. Diagnoses with B117 increased faster than did non-B117 diagnoses in the same areas. And index patients with B117 infections infected a higher proportion of contacts than index patients infected with other variants. And similarly, this graph here shows a similar rapid spread of the B1351 variant in South Africa to become the dominant strain. So let's move on now to the effect of the variants on disease severity. Well, In a briefing paper published on the 22nd of January and updated on the 11th of February, the UK government's new and emerging respiratory virus threats advisory group, or NERVTAG, highlighted several preliminary analyses showing a possible increase in the severity of disease associated with B117 compared with other virus variants. The NERVTAG report contained a total of 12 independent analyses, and I've just highlighted 3 here, which showed that compared to non-B117 cases, B117 cases had a higher relative hazard of death within 28 days of the test and a higher case fatality ratio and death risk ratio. As a reminder, this was based on S gene target failures as a marker of B117. And these were, largely population-based studies with limitations, and more data is needed. And of note, one of these studies has been published in the BMJ just this week, if anyone wants to do any further reading. To date, there has not been an effect on disease severity reported with the other 2 variants. So let's move on to the effects of the variants on vaccine efficacy. As you well know, thank you. As you know, there are currently 3 COVID-19 vaccines authorized by the Food and Drug Administration for emergency use. These are the 2 mRNA vaccines, Pfizer-BioNTech, and the Moderna vaccine, which are both 2-dose vaccines, and 1 viral vector vaccine, and that's the Johnson & Johnson single-dose vaccine. So evidence demonstrates that the authorized COVID-19 vaccines are both efficacious and effective against symptomatic, laboratory-confirmed COVID-19, including severe forms of the disease. Next slide, please. But sera from mRNA COVID-19 vaccine recipients, both Pfizer, BioNTech, and Moderna, have generally demonstrated modest reductions in antibody neutralization activity against a variety of mutations. Across studies, the greatest reductions were observed, followed by P1. Reductions for B117 were minimal. I've pulled out 2 such studies here, but there are plenty and more on their way. So in this first one, this in vitro study by Liu et al. Compared neutralizing activity of the Pfizer-BioNTech COVID-19 vaccine against a recombinant strain harboring the B1351 spike protein mutation. And you can see that compared to wild-type virus, neutralization of the recombinant B1351 spike virus was weaker by approximately 2/3. And in this in vitro study by Wu et al., assayed the serum neutralizing activity of specimens obtained from recipients of the Moderna vaccine this time against engineered pseudoviruses bearing the spike protein from wild-type virus, the B117 variant, and the B1351 variant. So on the left, you can see that the B117 pseudovirus added to the serum of persons who had received Moderna COVID-19 vaccine found that the pseudovirus had no effect on the vaccine's neutralizing ability. By contrast, on the figure on the right, there was a decrease in titers of neutralizing antibodies against the B1351 pseudovirus. However, it must be noted that across studies, antibody neutralizing activity of sera from vaccinated persons was still generally higher than that observed from convalescent sera from persons who have recovered from COVID-19. And it's difficult to predict how this reduced immunogenicity demonstrated by in vitro studies may impact sort of real world COVID-19 vaccine effectiveness, especially given that there are no immunological correlates of protection for SARS-CoV-2. So what can we say right now about vaccine effectiveness? Well, we have some evidence on the Pfizer vaccine. So preliminary results from the UK demonstrate that vaccination with 2 doses of Pfizer-BioNTech COVID-19 vaccine was highly effective against SARS-CoV-2 infection and symptomatic COVID-19 during a period where B117 was the predominant circulating strain in the UK. And similarly, high Pfizer-BioNTech vaccine effectiveness against infections was observed in Israel in the context of multiple circulating strains with a high proportion of B117 variant. And looking at the Johnson & Johnson vaccine, preliminary data suggests that this vaccine may have reduced overall efficacy against the B1351 variant. In the United States, efficacy was 72%, but in South Africa, where 95% of infections were due to B1351, efficacy was only 64%. However, Janssen vaccine efficacy against severe disease was high and similar across sites. So while vaccine efficacy against emerging SARS-CoV-2 variants remains under investigation, it does seem that preliminary evidence suggests that the COVID-19 vaccines presently authorized in the US will likely be effective against emerging variants. It's important in addition to remember that while vaccination efficacy is assessed on an individual level, vaccination itself is a public health intervention, and we need to consider this on a population level. There's a growing body of evidence that suggests that COVID-19 vaccines may also reduce asymptomatic infection and potentially transmission. And on a population level, high vaccine coverage can compensate for lower vaccine efficacy. What this means is that lower levels of vaccine efficacy may still be enough to protect the population if they're still widely distributed with a high uptake. So getting the vaccine into people is still the priority, and we need to increase vaccine uptake. So let's move on in the final couple of minutes to what CDC is doing regarding the variants. So the CDC, in collaboration with other public health agencies, is working to detect and characterize viral variants. We have staff available to provide technical support to investigate the epidemiological and clinical characteristics of SARS-CoV-2 variant infections. And we're also conducting an investigation of COVID-19 vaccine breakthrough cases, including those caused by the variants. CDC, next slide please, is also leading in tracking emerging variants through genomic surveillance. We're leading the National SARS-CoV-2 Strain Surveillance, or NS3 system, partnering with commercial diagnostic laboratories to sequence samples across the US, collaborating with universities and public health agencies to conduct genomic surveillance research, and supporting state, territorial, local, and tribal health departments. And finally, leading the SPHERES Consortium, which is a new national genomics consortium to coordinate SARS-CoV-2 sequencing across the United States. So with that, I'll end with a reminder to please wear a mask, stay 6 feet away from those not in your household, avoid crowds and poorly ventilated spaces, wash your hands, and get your vaccine. Thank you.
Thank you, Dr. Natarajan. Excellent talk. So now let's turn to the roundtable discussion. So I'll ask the panelists to turn your cameras on. Our previous speakers for this roundtable will be joined by Dr. Vinita Kumar, a professor of medicine and the medical director of the Incompatible Kidney Transplant Program and the lead nephrologist for the Living Kidney Donor Program at the University of Alabama in Birmingham. So we've had some very interesting questions from the audience. I want to start, Mary, with you. Dr. Dittrich, there have been several sort of practical questions that have been asked. Can you tell us some more about the infusion during dialysis? How does that work? How long is it? How do you set that up? Do monoclonal antibodies, do you think, do they stick to the dialysis membrane or not? Does the process cause any increased clotting? And then finally, just a quick question, do you use a PVC infusion set or the standard infusion set with the 0.2 filters?
All right, let me take some notes so I try and hit all those things. So let me start with the timing on dialysis. So we were prepared to use any of the products available, including the Regeneron combination, and as it comes available, the bamlanivimab etesevimab combination, but we got bamlanivimab. And so the initial way that had to be administered was a little complicated. You drew up 20 mLs of the medication, you wasted 20 mLs in a bag of 250 cc saline, then put the medication into the remainder of the bag and infused over 1 hour. The EUA has evolved on that such that now you can give it in a variety of quantities of saline, starting giving over 16 minutes in 50 mLs and then all the way up to the 250 mLs over 1 hour. So we give it in 250 mLs of saline over 1 hour. That's what we're doing right now. And we give it the 3rd hour of dialysis, or the hour before the last hour of dialysis. So if it's a 4-hour treatment, we give it hour 3 and observe hour 4. If it's a 3-hour treatment, we give it hour 2 and observe the final hour to comply with the requirements. Does the medications adhere to the membrane? No one knows. So we are operating in the absence of data here. What we do know is it's a large molecule. It's over 150 kilodaltons. So we have a lot of confidence after talking to experts, including our colleagues at the pharmaceutical companies, that it's not dialyzed off. So the statement in the EUA is that the PK is unlikely to be affected by dialysis, but there's this theoretic concern that it could adhere to the membrane, which is why we initially gave it after dialysis. But as I mentioned, that was so logistically challenging. So we conferred with experts and gave it on dialysis, and this is our thought. There's a theoretic adherence to the membrane, but no real reason to think that that would happen in any meaningful quantity, and it's better to get the infusion to the patient than forego it, especially with what we know or what we're seeing about its efficacy. So that's the decision we made, but the fact is I think we're operating in the absence of true data about that. But we're seeing good results, and logistically it's easier to give it on dialysis. There may be others on the call that have expertise or thoughts on that, but having talked to a lot of people, I think that's the best answer I can give.
Okay, and the question about the infusion set, does it have to be a special infusion set?
So the EUA states the infusion set, which is the one I described in my slide. And so we obtained that, easily obtained. You can get it from most of the distributors, and we had no problem getting it, and we used that. So. Okay. And there was a question about clotting. We haven't seen that, and I don't think that's been reported. We haven't seen accesses clot or issues that lead us to believe that clotting would be an issue here, and it's not reported in the side effects of the literature I've read. Dr. Anderson may have more thoughts on that.
No, we haven't seen a lot of clotting. I'm so impressed with both your presentation and your work, and just the notion of you've got the patient, they've got access, they've turned positive. I mean, that's just brilliant in trying to make a difference. And I think one of the reasons we were so excited to present to ASN is just trying to figure out how to propagate it. I think you did a very nice job of saying this is early data. This doesn't feel like the typical grand rounds that we're used to giving, but if we can do innovative things like you're doing more so across the country, then I think more patients are going to benefit.
Thanks. Dr. Natarajan, so one of the questions is, what are the data on natural immunity? How many reinfections? And if reinfected, are they mainly due to different variants?
So that's a great question, and that's something we're actively working on, and actually that's my day-to-day job currently within the CDC's emergency response is in the reinfections unit. So this is something we're really looking at trying to tackle at the moment. So we should have more data emerging in the coming days and weeks. I think there are lots of issues with the issue of reinfection and the variants regarding where you look and what the prevalence is of the variants in the countries you're looking at. So for example, in the UK currently, the B117 variant that I've spoken of is the dominant strain. So if you were to get reinfected in the UK currently, then the chances are you might get reinfected with the 117 variant, and whether that's because that's the circulating strain right now, or whether that's because the 117 being your immunity against it, the previous SARS-CoV-2 lineage you may have had is really interesting, difficult to tease out and under study at the moment.
Yeah, thanks. We'll tune in again for an answer once we have more data. Dr. Kumar, several people have really been interested in the use of monoclonal antibodies in transplant patients. Have you or your colleagues used monoclonals for transplant patients? Are they safe? Do they work?
Great, excellent questions, Dr. Kliger. So yes, both at our institution and colleagues across the nation have used monoclonal antibodies in our solid organ transplant patients. And while the EUA was not specific to organ transplant recipients, it did include immunocompromised state as defined by either a condition or from immunosuppression treatment. So that allowed these agents to be used in the transplant population. The question about safety, that may be somewhat easier to answer. The infusion-related side effects seem very similar to the general population and the dialysis population and generally well tolerated. Now, do they work? There's a wide range of anecdotal experience that's being discussed in the transplant circles. The experience is quoted anywhere from no response to significant and up to 50% reduction in hospitalization rates. And the difference probably stems from if it's a one-off, two-off, three-off cases, and they happen to get hospitalized after the infusion, they would assume there's no response to treatment. To groups that are seeing differences in treatment in hospitalization rates depends on what comparator group they're using. If it is a historic cohort of COVID-infected patients or COVID-infected transplant recipients who've chosen not to be treated. But all of it to say, what I would tell our audience is that if there is a benefit, the magnitude of this impact remains to be seen. And I ask our audience to stay tuned for data that is rapidly evolving, and a few centers have accumulated enough numbers of treated patients. So we may be seeing some publications come out in the coming time ahead that may shed some additional light. So I think I covered those three questions that you asked, Dr. Kliger.
Great, thank you. Dr. Anderson, are monoclonal antibodies safe in a patient who's been vaccinated for SARS-CoV-2? And would you give antibodies to someone who's been fully vaccinated, whether or not they have demonstrable antibodies?
Yeah, no data. So we're flying with little data up until this point, and now, at least as my read of the literature, we're flying with no data. I saw that question as we previewed. So I think, like everything, it's the risk and benefits of the therapy. If somebody has been vaccinated but now has turned positive and they fit in those risk factors, I'm not aware. And once again, this is Mike, the intensivist, as a contractor. I'm not aware of inherent, 3, 4, 5-fold increased risk of doing so. So if I've got a patient in front of me and they're in the high risk group and they, God forbid, turn positive after being vaccinated, I would strongly consider. But you're in even less data territory than what my wonderful colleagues have presented up until now.
Yeah, I mean, part of this challenge, and I just ask others to comment, is that we nephrologists and physicians in general have been evidence-based practitioners. And here we are in the midst of a pandemic. I mean, we remember our former president grabbing on some potential therapies for which there was little or no evidence. And here we are likewise in an area of only modest evidence. So how should we be thinking about that challenge different than the way we did a year ago?
I'll take a stab, and then my learned colleagues can either refute or support it. It's interesting. Once we get over this public health emergency, PHE, and get to write what I'm sure going to be 8 books on, 20 books on how to do this better, God forbid, for the next public health emergency, there really is an interesting lane of how should physicians react when a public health emergency is declared, an EUA is written. What does that really mean? Because I think we were building this aircraft as we were flying it with really smart people like the FDA and all the great physicians on this call. But I do think we, whether it's part of baking into a curriculum of residency, whether it's starting to, after this, gather more physicians, what does it really mean when a public health emergency is declared and an EUA medication comes out? Quite frankly, in my medical training, I got nothing in my training about this. So I don't know if it's a really good answer, but I do think one of the things we have to get better for handing the baton to the next generation is, what does this really mean for physicians who, of course, are evidence-based, who want to do things that are not harmful to patients and really have benefit but quite frankly, we're in the middle of a war. And how do we balance the risks and benefits of therapies? I think that's going to be a really interesting question that we can ask and answer.
So Mary, I wonder what your thought is. I mean, you're a leader of physicians with a whole, cadre of doctors looking to you for policy and advice. How do we think differently now, or how do you think differently now than we did a year or 2 years ago?
That's actually a great question. And as I mentioned, I take care of my own large panel of dialysis patients as well. So I see this both as someone who is in a leadership position and responsibilities therein, but the greater responsibility in many ways is to the patients I care for. And one thing I would say, I don't have particularly unique insights, but what I will say and how I think about this is not using this therapeutic is not a risk-free choice. We know what happens when we don't treat these patients. The data is clear. They go in the hospital, they get ill, and many of them die, and they have long COVID. So it is not like the risk-benefit has a bad risk of not treating. So therefore, the risks of any novel therapy become more acceptable. So that's how I think about it. And I read the early data, and we were acting without data, but we were acting without other options. And so I felt more comfortable, given all those considerations, trying this therapy, given that monoclonal antibodies have been around forever. I mean, we use Rituxan, we use a lot of these. So that's how I thought about it, and that's how I talk to my colleagues about it. And again, in our population, doing nothing has bad outcomes.
Yeah, really helpful. Can I just ask Commander Foss, I mean, you're a pharmacist by training, and now in the business of propagating this, have you changed the way you think about these kinds of challenges?
Yeah, I mean, pretty much what Dr. Dittrich was saying. It's just like the whole evidence-based things, like pounding that, and if there were doctors that went with anecdotal, pounding in the evidence-based treatments, and now it's kind of like opposite, because like Dr. Dittrich said, we just don't have any other options. So this is really the best option we have right now. So I pretty much just mirror everything that Dr. Dittrich says, and agree completely.
Well, let me ask either you or Dr. Anderson, how are you assuring accessibility of these therapies to rural and lower socioeconomic communities?
Well, I know for lower socioeconomic communities, we are federally qualified health centers are one of our pillars, one of the 4 pillars that we do provide to, so that they do a great job serving the lower socioeconomic classes. As far as rural communities, I'll defer to Dr. Anderson on that one.
Dr. Kliger, if I could just add a comment about that. One way they're ensuring adequacy or adequate distribution is to give it to dialysis patients, 50% of whom are, people of color and certainly lower socioeconomic classes or have a high representation in our patient population. So their work with us gets this medicine to people who are disadvantaged through a variety of social reasons as well.
So, Dr. Anderson, I don't know if you heard, I wonder if you could make a comment about availability in rural or lower socioeconomic communities. How do we?
I apologize about being late. There was a knock at my door, which I don't expect in a federal building because there's very few people here. So we are re-energizing our efforts around health equity. And I just want to personally and professionally thank the Biden administration. There's a new energy. There's an Office of Health Equity for COVID-19 run by 2 amazing internists, Dr. Nunez-Smith and Dr. Webb. They are in constant communication with the federal team saying, it's terrific that we're starting to see a thaw in the nation, but and we know there are thousands of patients that can still benefit from this for all the reasons the panel has outlined. So I don't want to get ahead of my skis, but suffice it to say between now and Monday, I think you'll see some renewed energy out of the White House and Health and Human Services of making sure that we partner with FQHCs, dialysis centers, urgent care centers. I mean, there are so many potential places to reach patients that remain untapped. It's core to who we are as leaders in this health and human services space, and I think you'll be really impressed how the administration is putting a lot of energy behind it.
Yeah, thanks. Dr. Natarajan, how stable are the current variants compared to the SARS-CoV-2 virus sequenced earlier last year? Should native antibodies be checked in all patients admitted with coronavirus?
Well, we're currently doing genomic surveillance to understand better the distribution of these currently circulating variants. New variants emerge, some persist, some disappear. So the variants that may affect vaccine efficacy are currently very low prevalence. So I think it's difficult to answer that question. In regards antibody testing, there aren't any specific guidance related to antibody testing recommended to address the variants. So again, I think there's an absence of guidelines there.
Yeah, so as in so many other pieces of what we do, we're still flying by the seat of our pants, trying to sort of figure out. I mean, actually one of the secretaries who works in our office had the disease, is now going to be getting her first of the vaccines 90 days after she had the disease. And she asked me the question the other day, well, should I be checking my antibody levels? Would it be at all helpful to know what they are? I've heard that a second dose in someone who's had the native disease has in some cases been very difficult with lots of, reaction because of high antibody, well, because of previous exposure, whatever the reason. But I wonder what any of you think about that, about antibody testing in this population of patients.
I don't know what it gets you. I don't know how it guides next steps. And maybe I'm just out of my comfort zone here, but I think that as many people can get vaccinated as possible. And it's just a personal anecdote. I've had a couple of friends who are in the same instance that they've had the disease. They're better now. They've had their first dose, but they've been reading in the lay press and a couple things that, is the second dose really a good idea? And I haven't seen official guidance not to do it. So a slightly rambling answer to, I don't know what that antibody profile is going to help you with.
I would probably add, I agree with Dr. Anderson. What I would add is also that there are multiple different testing platforms. We don't know which antibodies are truly neutralizing antibodies. We don't know what is that threshold beyond which, if they are protective, what would that threshold be? So in the absence of clear information, checking for antibodies doesn't seem to add any additional clarity to what we know now. So I would say, unless it's part of a research trial, we're advocating for our patients to not get tested for antibody.
Yeah. So Vanita, you're an expert in the immune system. So let me ask you this question. Many have wondered what role the cell-mediated immunity, T-cell responsiveness, is in this disease, and not just the antibody response. What can you tell about either data or your speculation about that?
So that's a very, very intriguing question. At the beginning of the pandemic, pretty much most centers decided that they would reduce immunosuppression to augment T-cell responses to help eradicate or reduce the acute viral phase of the infection. Interestingly, some early data is emerged, and we don't know the generalizability of this data, but some early data suggests that perhaps reducing immunosuppression may not be the wisest, especially later in the course of the disease, where it's no longer just the acute viremic phase that's causing the problems, but it's the immune response to it. So the real answer is we don't know, but there are certain groups that are actively looking at the role of T-cell immunity in this process. And I think we should be getting more data in the next 6 months on that.
Yeah, thank you. Mary, let me ask you this, and also Michael, maybe the two of you can address this. What, if any, differences have been seen in efficacy and safety of the monoclonal antibody therapies comparing dialysis patients to the general population?
So I'll start, and this is easy. There's no data. How many times have we said that? So the data that I showed you today may be the first sort of public distribution of data on dialysis that I have seen. Now, as I mentioned, I collaborate very closely with George Aronoff at DaVita and Jeff Himes at Fresenius, and we share our experience, but there's no published data on this. Again, I'll go back to there's no obvious reason why safety or efficacy would be different in this group of patients, other than they may benefit more due to the high high risk category they're in. And there is some thought that dialysis patients have an impaired immune response, and so passive immunity may the passive acquisition of antibodies against this to prevent viral cell, the virus entering the cell and replicating may be beneficial, but we don't know. Dr. Anderson?
Yeah, I'll add, I have now seen the data on dialysis patients getting the antibodies, and like David said, we're encouraging clinicians not only to help us get these therapies into patients, but publish your results. And I think that whatever energy we can get behind, pardon me, more and more of our colleagues putting their experience out is terrific. I know of papers coming out of New York in short order. The state of Michigan has coordinated a massive effort. Intermountain Health in Salt Lake is putting a publication together, as I understand it. We're putting one together of the DMET teams that we put together in 3 locations. So I would encourage people to publish. I'm gonna take an audible for a second and relay one thing. That is the variants and the antibodies. And I don't think, and I apologize, I had a visitor, I don't think I heard in the questions. The notion of with variants, are these antibodies as effective as we think they are based on the hour we've just presented? I would encourage you over the next 4 or 5 days to look to announcements from the FDA about the Lilly BAM alone antibody and the quote unquote California virus. I'm sorry, California variant. And my CDC colleague can give me the numbers that I never get correct. But that points to good news for me, actually, in that we are being in very real time assessing these therapies versus the variants that are emerging and getting data to clinicians as soon as possible. So I would be remiss if I had wonderful colleagues on the line and didn't share the fact that the BAM product alone from Lilly versus the quote unquote California variant, I think you're going to see something from the FDA and CDC pretty soon on that.
What a teaser. Thank you, Michael.
I don't mean to be, I'm so sorry.
So Vinita, let me ask you this question. Are there any issues if you're called for a transplant after monoclonal antibody infusion? Patients are concerned, maybe, I don't want to jeopardize my possibility of getting a matched transplant.
Absolutely. That's what a great question. Are there any issues if you're called in for transplant after a monoclonal antibody infusion? So, we've all started our answers by saying there's no data. And I liked what Dr. Anderson said earlier about how does organized medicine respond in the face of lack of data. And in that situation, I say I want to bring clarity to the confusion that exists, if you will. So there are a few things we can actually talk about pretty well. In theory, infusion with these monoclonal antibodies should not be of concern in forming new HLA antibody. Why is that? Because these are monoclonal products made in a lab. They're binding monospecifically to certain proteins, and in this case, SARS-CoV-2. And so they should not theoretically have HLA cross-reactivity. And we know from use of other monoclonal agents post-transplant like rituximab or Campath or adalimumab that's used for RA or psoriasis, or infliximab for Crohn's disease in transplant patients, and they do not seem to incite an anti-HLA response either pre or post-transplant, and therefore it should not adversely affect chances of future transplant if you get called in for a transplant. The word of caution that I would like to share with our audience is that these infusions are, after all, IgG particles, and that can interfere with antibody binding in vitro and affect the interpretation of the HLA data. So the takeaway for our dialysis providers is, if you have a patient on dialysis that is getting this infusion, is on the transplant list, to let your HLA lab or transplant center know so they can treat the sample with this knowledge and interpret the results accordingly. And then the final word of caution is, I frequently hear monoclonal antibody infusions sort of confused with infusions with convalescent plasma. That's a completely different thing. And convalescent plasma may have a different implication in terms of HLA adverse implication, but monoclonal antibodies, we think, don't.
Thank you. So one last quick question. Do we know if Eli Lilly and Regeneron are going to apply for BLA for full FDA approval?
I understand that's the path forward. And I will say also, I think and I'm sorry to take up more time, Dr. Dittrich did a very nice job of saying, by the way, at first these antibodies were being distributed by the states in sort of a wartime footing, and now we've really moved to direct ordering. I think that's a more efficient way for a dialysis center or a hospital to get it. It is the hope within the federal COVID response that these therapies, I keep saying, the federal government doesn't order ceftriaxone, right? It becomes a part of the fabric of modern medicine. As we move to more FDA approvals and we move to more direct ordering, then academic medicine and the normal processes take over. And that's really our hope.
All right. Well, let me wrap it up then. We're close to the end, and I always like ending a little early because giving a minute or two back is better than going over. And this has really been a wonderful hour and a half. I'd like to really thank the speakers for your talks. But in addition to that, for a really lively and interesting roundtable discussion, I think we really delved into it really nicely. If anyone has any additional questions, please contact the ASN and we'll try to direct them to the right people. On behalf of the American Society of Nephrology, its kidney community partners, and all of our presenters, thank you for joining us and participating in this roundtable discussion. Have a good day, everybody.